Do you know what is the meaning of paper chromatography? At first, chromatography means a technique used to separate different components of a mixture. By using chromatography method, we can separate different components of a mixture. In case of paper chromatography, we use a paper, cellulose paper, and where we will cut this paper like this, and we will put our sample here. Then we will put it in a jar or setup where solvent is placed at the bottom, and we will place that paper in such a way that the tip will dip inside that solvent. Then what is that solvent actually? That solvent consists of two parts, one aqueous part which consists of water, and one organic part which is non-polar, and that aqueous part is polar and organic part is non-polar. Aqueous part bind with the paper and act as stationary phase, and non-polar part move upward and will reach up to a certain point called solvent front. This non-polar solvent by taking this sample will move upward, and some components are polar and some components are non-polar. More polar solvent will move slower, where non-polar part will move faster. And we can also calculate retention value or RA value, which is by the formula distance travelled by the solvent. This is the distance travelled by solvent, and distance travelled by a particular solute. So A divided by B. So it is the retention value.